Hello, what's going on, Gracians? Welcome back to another episode of The Scientific Mind. Today we are going to talk about cellular respiration. But before that, let's have a prayer first. Okay, so if you are ready, let's start the discussion. Cellular respiration, a food utilization process. On the previous episode, you have just learned how plants produce foods for consumers like you and for their own consumption. The food they produce will make the life of all those that are dependent on plants possible. In order to harness the energy from food, consumers would also have a specialized process. However, not all living organisms need air, such as anaerobic bacteria, but all of them need a food to extract energy through a process called cellular respiration. Heterotrophic organisms like humans use catabolic processes to extract energy. In catabolism, glucose is broken down into smaller and more useful compounds. Cellular respiration involves the catabolism of glucose along with oxygen to produce energy. Carbon dioxide and water are its byproduct. So what is a cellular respiration? Cellular respiration is a metabolic pathway that breaks down glucose and produces ATP. Cellular respiration is an energy-releasing process that takes place in all active cells 24 hours a day. It starts in the cytoplasm and is completed in the mitochondrion. There are two kinds of cellular respiration. First is the anaerobic respiration, which does not need oxygen. Processes included in the anaerobic respiration are glycolysis and fermentation. The second kind of cellular respiration is the aerobic respiration. It needs oxygen to produce ATP and it is more efficient than anaerobic. Processes includes glycolysis, Krebs cycle, and oxidative phosphorylation. Glycolysis. Glycolysis is the breaking down of glucose into two molecules of pyruvic acid. Glycolysis takes place in the cytoplasm of the cell and does not require oxygen. It is said to be the first energy harnessing process during the primordial times, when no oxygen gas was present in the Earth's atmosphere. During glycolysis, two phosphates in the form of ADP or adenosine diphosphate are added to a 6-carbon sugar molecule and then split into two 3-carbon sugars, which is glyceraldehyde 3-phosphate or the G3P. During glycolysis, some hydrogen, energy, and water are removed from the G3P, eventually producing two pyruvates. Nicotinamide adenine dinucleotide, a hydrogen acceptor which becomes reduced to NADH, is a cofactor in transport task to pick up hydrogen ions and electrons released during glycolysis. All in all, Glycolysis is a glucose splitting reaction. It is a process in which one molecule of glucose is broken down to form two molecules of pyruvic acid. It occurs in the cytoplasm of the cell. 4 ATP and 2 NADH are produced, but this pathway produces a net gain of 2 ATP per molecule of glucose. To summarize, there are three changes that occur during glycolysis. First, glucose is converted into two molecules of pyruvate. Second, two molecules of ADP are converted into ATP through substrate level phosphorylation. And last, two molecules of NAD plus or nicotinamide adenine dinucleotide are reduced to NADH. Okay, so next process after the glycolysis is the Krebs cycle. Following glycolysis, the mechanism of cellular respiration involves another multi-step process, the Krebs cycle, which is also called the citric acid cycle or the tricarboxylic acid cycle. The citric acid cycle is a series of chemical reactions that takes place in the mitochondrial matrix. This uses acetyl-CoA derived from sugar and fat breakdown to form ATP, NADH, FADH2, and carbon dioxide. The NADH and FADH2 can be used to form additional ATP through the electron transport chain. The citric acid cycle goes by many names, including tricarboxylic acid or TCA cycle and the Krebs cycle. Citric acid refers to the citrate that is produced in the first step of the pathway. The tricarboxylic acid title gets its name from the 3 carbon dioxide that are produced for each fully oxidized pyruvate. Krebs refers to Hans Adolf Krebs, who identified the full cycle in 1937. He was eventually awarded the Nobel Prize for Physiology or Medicine in 1953 for his discovery. In the first step of the cycle, an enzyme called citrate synthase joined the 2-carbon acetyl group from the acetyl-CoA with the 4-carbon oxaloacetate to form 6-carbon citrate. In step 2, an enzyme called aconitase converts citrate into isocitrate. Next, an isocitrate dehydrogenase enzyme oxidizes isocitrate, a 6-carbon molecule, to a 5-carbon alpha-ketoglutarate. The carbon that was lost is released as carbon dioxide and 1 NADH is produced. The carbon dioxide that is released was originally part of the oxaloacetate and not acetyl-CoA. In the fourth step, an enzyme called alpha-ketoglutarate dehydrogenase converts alpha-ketoglutarate into 4-carbon succinyl-CoA. Similar to step 3, this reaction produces 1 carbon dioxide and 1 NADH. In step 5, a succinyl-CoA synthesase enzyme converts succinyl-CoA into succinate. This produces GTP, which is converted to ATP. In step 6, an enzyme called succinate dehydrogenase converts succinate into fumarate. These steps makes FADH2. In step 7, the fumarate hydratase enzyme then converts fumarate into malate. In the final step of the citric acid cycle, a malate dehydrogenase enzyme converts malate back to oxaloacetate. Like all steps involving a dehydrogenase, a coenzyme is produced. Here it is a NADH. The oxaloacetate that was generated through the citric acid cycle is now ready to join the another acetyl group and begin the cycle a second time. For every 1 glucose that is broken down through glycolysis, 2 pyruvates will be produced. These 2 pyruvates will produce 2 acetyl-CoAs. For every 1 glucose, 2 acetyl-CoAs will be made and 2 turns of the citric acid cycle will occur. This means each product of the cycle must be doubled. A total of 4 carbon dioxide, 6 NADH, 2 FADH2, and 2 ATPs are made through the citric acid cycle. NADH2 and FADH2 are electron carriers that can produce more ATPs later in aerobic respiration. In addition to sugar like glucose, proteins and fats can also provide carbon substrates to fuel the citric acid cycle. Proteins can be broken down into individual amino acids such as alanine, aspartate, and glutamate and converted into intermediates in the cycle. Fatty acids can be broken down into acetyl-CoA, which then begins the citric acid cycle. This metabolism of sugar, proteins, and fats through the citric acid cycle provides vital energy necessary to maintain many cellular processes. The next process involved in the cellular respiration is the oxidative phosphorylation. Electron transport chain is a series of proteins and organic molecules found in the inner membrane of the mitochondria. In this part, electrons are passed from one member of the transport chain to another series of redox reduction. Energy released in these reactions is captured as a proton gradient, which is then used to make ATP in a process called chemiosmosis. Together, the electron transport chain and chemiosmosis make up the oxidative phosphorylation. So what are the key steps of oxidative phosphorylation? First is the delivery of electrons by NADH and FADH2. The reduced electron carriers, NADH plus FADH2 from other steps of cellular respiration, which is the Krebs cycle, transferred their electrons to molecules near the beginning of the transport chain. In the process, they turn back into NAD plus and FAD, which can be reused in other steps of cellular respiration. Second step is the electron transport and proton pumping. As electrons are passed down the chain, they move from a higher to a lower energy level, releasing energy. Some of energy is used to pump hydrogen ions, moving them out of the matrix and into the intermembrane space. This pumping establishes an electrochemical gradient. Next step of the oxidative phosphorylation is the splitting of oxygen to form water. At the end of the electron transport chain or ETC, electrons are transferred to a molecular oxygen, which splits in half and takes up hydrogen to form water. Next step, gradient-driven synthesis of ATP. As hydrogen ions flow down their gradient and back into the matrix, they pass through an enzyme called ATP synthase, which harnesses the flow of protons to synthesize ATP. The electron transport chain. Electron transport chain is a collection of membrane-embedded proteins and organic molecules, most of them organized into 4 large complexes labeled as 1 to 4. In eukaryotes, many copies of these molecules are found in the inner mitochondrial membrane. For prokaryotes, the electron transport chain components are found in the plasma membrane. So what is the important function of ETC? First, it regenerates electron carriers. NADH and FADH2 pass their electrons to the electron transport chain, turning back into NAD plus and FAD. This is important because the oxidized forms of these electron carriers are used in glycolysis and citric acid cycle and must be available to keep these processes running. Second, it makes a proton gradient. The transport chains build a proton gradient across the inner mitochondrial membrane, with a higher concentration of hydrogen in the intermembrane space and a lower concentration in the matrix. This gradient represents a stored form of energy, as well as it can be used to make ATP. So that ends our episode for today. This is Sir Eman of the Scientific Mind. I hope you learned some new knowledge about the topic. See you on next episode.